as infections, tobacco, ionizing radiation, pesticides, and diet. We know that these factors are present in all across our population. There's only some people who get leukemia, most people who do not. So genetics plays a big role in that. We see evidence of these causes in the mutational profile of leukemia itself. So we can see mutational signatures of the influence of environmental causes. We see gene deletions. I'll talk about that later. And also DNA methylation changes. I'll talk about that later too. So when I first started working on leukemia, we asked the question, what is the critical time windows for leukemogenesis? In other words, when in a child's life do these mutations occur? So we got one important clue about timing from twins. And here's an example of a twin. In this case, both of the children got leukemia. So this was a pair of girls, and one got leukemia at age 5, and the other got leukemia at age 13. So not very young, you know, the 13-year-old is pretty old for childhood leukemia. But interestingly, both of these twins shared the exact same translocation between a gene called TEL and another gene called AML1 in chromosomes 12 and 21. So what this proved to us, here's the translocation here, the two girls, age 5 and 13 on the right. This translocation, since it's exactly the same in both girls, it must have occurred before they were born, because at that time period, they had a shared circulation. So the translocation would have happened here prior to birth and was shared between the girls. It also tells us it takes a long time from that initial translocation for leukemia. It can take a long time for it to occur. So there must be other genetic changes after birth as well. So it turns out this is not only something we can do in twins. We've been able to look at a lot of singleton children and do what we call backtracking. That's looking back to the time of pregnancy by examining a neonatal blood spot. So these blood spots are taken from children right at birth. So we know that whatever we can look at in the blood would have happened prior to birth. And here's an example on the left-hand side of this slide where we sequenced a translocation in leukemia cells and then looked back on the blood spot and found that that translocation was already there at the time of birth. So we've been able to look at the many different mutations, many different translocations or mutations on the left-hand side here, and found them in the blood spots of children who got leukemia later. There are other genetic changes in leukemia that happen postnatally or after birth. An example on the right here is RAS gene mutations or gene deletions, such as this second hit 12p deletion here. I just wanted to see if I can get a, can you see my cursor? Maybe you can, so I can point at things.
Yes, we can.
Okay. So in general, we think of leukemia as a two-hit disease. The first initiating lesion occurs before birth, and the second mutation occurs close to the time of diagnosis. And that means that there can be two different causes. One cause or one factor would happen here before birth, and then there's some kind of a second hit later on. It also tells us that if we can identify these children who have these initiating lesions, maybe we can prevent the second mutation from occurring. So leukemia may be a preventable disease. We know that there's a lot more children who get initiating lesions, about 1% of children have pre-leukemic mutations at birth, but only a tiny percentage of them will have the second hit. And it may be that we can prevent leukemia by modifying the environment for children who have these initiating mutations. So what are the environmental causes or environmental factors? One big one for leukemia is infection. Leukemia is a cancer of the immune system. So this is the primary hypothesis of how infection may impact childhood acute lymphoblastic leukemia. We think that leukemia may be a response from changing our environment, our early environment for children, in a way that's not conducive to a healthy development of their immune system. What children need is early antigen exposure. Our immune systems are primed to interact with our environment. That will then lead to a trained immunity of the immune system, immune competence, and health for that child. But what's happened a lot for children in modern society is we delay that antigen exposure. So children may be in too clean of an environment in their first year of life, end up with dysregulated immune responses when they interact with the environment later on, and that will cause expansion of pre-leukemic clones. So from epidemiology, we have a few observations that support this idea. One is when we look at organized daycare attendance. So this is bringing a child to a formal place where they are looked after by a childminder. This is actually healthy and reduces risk of childhood leukemia. So we see many different studies here that have examined exposure to early daycare and found that there's overall, the odds ratio here is 0.77, indicating that daycare is protective for children. But I do want to point out one of these studies, it's a very big one, Gilham 2005 here. This is the United Kingdom Childhood Cancer Study. So it's got a big square. Its daycare is very protective here. But this same study also was able to look at general practitioner records for early physician diagnosed infection. And interestingly, the cases here, whoops, I didn't label it, but the red line here are cases and the green line are controls. You can see that cases have a higher rate of early physician diagnosed infection than controls. So this is kind of interesting. If going to daycare is protective, don't children get infections in daycare? Yes, they do. But the children who end up in the doctor's office for infections actually have a higher risk of leukemia. The children who respond to infections in a more healthy fashion and don't go to the doctor, they end up with a lower risk of leukemia. So this brings up another question. Are children who get lymphoblastic leukemia, do they have different immune systems right at birth so that they're actually responding to infection in a different way? So to investigate that question, we in California have some resources which are very helpful. These neonatal blood spots, which I've already introduced to you, are available for over 23 million children born in California since 1982. So we are able to access these blood spots and do research. And we are also able to examine if there are any differences in development of children who get cancer compared to those who don't. So we took that opportunity to examine cytokines in these blood spots in cases children who got childhood leukemia, 116 of them compared to controls. So this is comparing 116 cases to controls and looking at cytokines, which are chemical messengers of the immune system. We found that there was one cytokine which really was profoundly different in children who grew up to get leukemia, and that's interleukin 10. You can see these odds ratios here are quite extreme. So when we compare the group with the highest levels of cytokines, we find a 0.04 odds ratio, which means about a 20-fold difference between the cases and controls, and a profound deficit of interleukin 10. Interleukin 10 is an immunosuppressive cytokine. So if it's low, it would make sense that a child who got an infection would end up responding too strongly to that infection. They would show a very fulminant infectious disease, which may be damaging to their immune system. So the question is, what causes this profound deficit of interleukin 10 at birth? Could it be that is it genetic? Or is it involved with some environmental exposure during pregnancy? So we thought that it might be that they might have an infection that occurs before birth that alters the development of their immune system. And to find such an infection, we sequenced RNA from children with lymphoblastic leukemia. And we know that children who have lymphoblastic leukemia have depressed immune systems, so they're going to have they may show infections more commonly than controls. So to compare we compared lymphoblastic leukemia to myeloid leukemias as a control. And the reason for doing that is AML has no infectious etiology like ALL, but the patients are immune suppressed. So both groups of lymphoblastic and myeloid leukemias should show infections in their diagnostic cells, but we were looking for something specific in the lymphoblastic leukemias, and we found something specific. And what that is a virus called cytomegalovirus, or CMV. So CMV, and these are in very young children. We found a lot of CMV in leukemia cases, and we didn't find it in any myeloid leukemia cases, even though these myeloid leukemia cases tend to be older children. And this is at the individual level here. Amongst these 36 ALL cases, we found active CMV viruses in about 2/3 of them, and very rarely amongst some AMLs. Then the idea here, we also wanted to see if CMV was present from the time of birth and perhaps causing this low IL-10 profile that we saw. So here we used a very sensitive PCR assay called droplet digital PCR, or ddPCR, to detect CMV at the time of birth and compare with the hypothesis that children would more commonly have CMV who got leukemia compared to controls. And that's what we found in this initial study. So we found that children were more likely to be CMV positive, about 4-fold more likely. And this was even more common in Hispanics, so children of Central or South American ancestry were more commonly positive with CMV than their controls. And Epstein-Barr virus, another virus known to be causal for some hematopoietic malignancies, was not any different between cases and controls. Looking at a population level, this is from the Swedish registries. So in Sweden, with National Healthcare Service and careful record keeping, the investigators there are able to examine the infection status of pregnant mothers and children at birth from leukemia cases compared to controls. And in this analysis, it was more than tenfold more likely to have CMV diagnosis by a physician in children who got lymphoblastic leukemia compared to controls, indicating that CMV is very likely a major risk factor for lymphoblastic leukemia in children. Not CMV just by itself, but having an early CMV infection is key, because we all end up getting exposed to CMV in pretty much worldwide at some point in our young life. But really, it's only if we got it during pregnancy or around the time of birth. And the question is, what might be going on? Is CMV causing this low IL-10 phenomenon that we observed? It's very possible that it is. So in this study, this is a small number of children, some who got leukemia and some who didn't. We compared mom's second trimester cytokines with children's cytokines at birth, and also looked at CMV proteins in children, and including a gene called, or a protein called CMV IL-10. This is a version of interleukin 10 that cytomegalovirus carries, and it turns out it's inversely associated with many of the cytokines from mom. We haven't looked at CMV in mom yet. We're doing that right now because we want to see whether CMV in mid-pregnancy for mom is what's driving the interleukin 10 result in children. But the hypothesis here is that the virus-derived IL-10, CMV IL-10, is inversely associated or suppressive for human IL-10. So that could be the cause of the low IL-10 at birth. Okay, I think I'll just go through this quickly just to say what CMV may be doing. We found that there are differences in the association with CMV in leukemias, depending on the leukemia subtype. And so what we found is that children who got leukemia, which is high hyperdiploid, commonly has CMV, but it's the children who have other subtypes besides high hyperdiploid, which tend to have stronger infection history, so that CMV may set up the immune system to be more responsive or sensitive to CMV in children who get leukemia of other subtypes. So just to summarize this section of the talk, the one thing we found that's really critical is these low IL-10 levels in children who get lymphoblastic leukemia. This may be a result of CMV infection. There are other potential causes as well, such as maternal fetal genetic interactions. This sets up a child to be more responsive to infections in a deleterious way, a damaging way that will then lead to childhood lymphoblastic leukemia risk. So the responder status at birth is the critical point here. Responder to infection. Okay, in the next section here, I'll talk about tobacco. So tobacco, we know, is a damaging exposure, contains a lot of carcinogens. But interestingly, there's not an overall association between tobacco and lymphoblastic leukemia risk. So here, if we look at maternal exposure to tobacco, it's a one-fold odds ratio. This is using data from the birth registry, looking at children who then contracted cancer afterwards compared to controls. And lymphoblastic leukemia, this risk is even lower, but notice this study is not very big. It's only looking at 8 children with leukemia. There are some cancers in children which are seem more strongly associated with tobacco exposure, such as gliomas perhaps, and also retinoblastomas. However, the story is a little different when we look at paternal smoking. That's smoking from the father. Now, in the US, paternal smoking is much more common than maternal smoking. So men smoke more commonly. So the rate of paternal smoking is about 30%, and maternal smoking only about 5%. So here, paternal smoking, which occurs both before and after pregnancy, the more a father smokes, the higher the risk of leukemia in the children. So how might paternal smoking be affecting risk? There is an interesting clue from a study. This was published way back in the 1990s, 1998, that looked at mutations in a gene called HPRT. And what they found was that when they had mutations in mothers who were exposed to tobacco smoke during pregnancy, those mutations were much more commonly VDJ recombinase mediated. So this is VDJ recombinase is a way that immune cells, B cells and T cells, will rearrange their immunoglobulin or T cell receptor genes. Whoops. And the VDJ recombinase does not normally cause mutations in HPRT, but it may cause off-target mutations at this gene, and it can possibly do this anywhere in the genome. And these mutations seem to be enhanced in the presence of tobacco. So here's a diagram of VDJ recombination. So this is on chromosome 11 here, where we have the IGH gene. It rearranges itself to create here the VDJ cassette, which makes an antibody to be have its own particular unique ability to react to antigens. So this is a normal process over here on the left, and on the right we see the mutations that the nucleotide sequence that signals VDJ recombination. This should only occur in B and T cells, but it can also happen at other parts of the genome. So our hypothesis with tobacco is that tobacco may cause this or enhance the presence of this off-target RAG recombination or VDJ recombination, and that this may induce gene deletions. Now, why are we interested in gene deletions? Gene deletions are very common in lymphoblastic leukemia, and they always occur after birth. So to look at gene mutations or gene deletions, we used an assay called MLPA that we could look at this list of genes here. These are genes that are commonly lost or deleted in childhood leukemia patients. And what we found is that in families where there was the presence of tobacco smoke in the child's environment, there was a much higher presence, that's the blue line here, of deletions present in the leukemia cells compared to leukemias that were from children that were not exposed to tobacco smoke. So here the risk ratio is 1.31. And when we looked at what point in pregnancy that exposure was most important, it's when the child was breastfeeding. So that odds ratio was 2.11 for if there was tobacco smoke present in at the time of breastfeeding for the mother. So this links tobacco to these postnatal mutations, these gene deletions. So earlier in my talk, we talked about prenatal mutations, the translocations, but here the key point in the child's life is postnatal. When the deletions occur, that's when tobacco smoke presence seems to be the most critical. More recently, we have sequenced the whole genome sequencing in children with leukemia, comparing those that got leukemia in a family that had tobacco smoke present compared to leukemias where there was no presence of tobacco smoke. And interestingly, we found the presence of these RAG sites, which the off-target RAG sites were much more common at the deletion breakpoints in the children who were exposed to tobacco smoke here, compared to ones who had low or no exposure. And these RAG sites were exactly at the point in the deletion. The distance from the breakpoint was exactly where you would predict if VDJ recombination was the actual cause. So I think this kind of proves the point at a molecular scale that tobacco is involved in causing deletions present in a child's leukemia cells. Okay, so now I'm going to switch a bit to genetics and genetics around the whole genome, and ask the question: are some children more susceptible to tobacco than others? Because we know that tobacco smoke is very common, but only very few children get leukemia. So here we used a GWAS approach. So this is from a study where we looked at 4,000 children who got leukemia in California compared to 4,000 controls. Here are 5 genetic risk factors which came out of this study. These are genetic risk factors which are used for making polygenic risk score for childhood leukemia. Genes such as ARID5B, IKZF1, PIP4K2A, CEBPE. So what we did then with this GWAS study is we added tobacco to the equation, looking at risk. And what we found is a much more complicated profile in adding tobacco. So basically, what I mean by adding tobacco is adding tobacco as an exposure across this entire population of 8,000 people. We found a whole bunch of other genetic risk factors. So this indicates that our polygenic risk score for childhood leukemia, which without the presence of an environmental factor is fairly simple. It's got those 5 genetic risks. If once we add an environmental risk factor, genetics becomes more complex, because here we are finding that some children are more susceptible to tobacco than others. So these 5 more genetic factors then contribute to a much more complex profile of genetics and childhood leukemia risk. So ARID5B does not interact with tobacco at all. So our G by E interaction over here is basically not significant. So this is a gene-only effect. So is IKZF1, CEBPE, and PIP4K2A. Now, some of these other genes have an effect on, or an interactive effect with the genes, or a disease gene effect over here, including like LRRK1, CDX1. These have low p-values over here, and other genes such as XOC3 has a strong environmental genetic effect, and then contributing to this 3 degree of freedom test here. So we ended up with 10 risk factors with the presence of tobacco. So the takeaway from this section of my talk is tobacco is an important risk factor for childhood leukemia. Even though it doesn't show up with maternal exposure, it seems to be paternal, and postnatal household exposure is important. And we have some very strong mechanistic effect that tobacco induces a specific mechanism with VDJ recombination to cause gene deletions. So this is a rare connection between exposure and a specific mutation, where we can see an environmental risk factor leading to a precise mutational mechanism in the tumor. And we also know that some children will be more sensitive to this mutational mechanism than others because of genetics. So now a bit about diet in childhood leukemia. And for diet, we have strong epidemiology evidence that children have a lower risk of leukemia when their family, their mothers, take prenatal vitamins compared to controls. And here's from our California childhood leukemia study showing that mothers who have a high intake of vitamin supplements during pregnancy have a much higher reduced risk of leukemia in their children and offspring. So we wanted to know what the mechanism of this nutritional benefit was. Could it be that exposure to folate at conception or in early pregnancy causes epigenetic switches in specific genes that lead to reduced risk? So for this part of the study, we looked at cases and controls again from California using the blood spots again and using a DNA methylation array. Now, there are some other natural experiments that have shown that nutrition is involved in DNA methylation. Nutrition during pregnancy is involved in DNA methylation of the offspring. And this is one of the most interesting studies, I think, and what I mean by natural experiment. In this study in Gambia, Africa, this is a study from Andrew Prentice, and what he did was to collect specimens from children at birth that were conceived in different seasons. So, in children who were born in the what they call the wet season, and compare them to children born in the dry season. Now, in Gambia, during the wet season, the availability of fresh vegetables that would contain high levels of folate is limited during the rainy season, but they are abundant during the dry season. So in comparing children who are conceived during the rainy season compared to those in the dry season, what these investigators found was that there was one particularly sensitive gene called VTRNA2-1, which was highly associated with the presence of healthy diet during close to the time of conception. And here you can see the difference in DNA methylation between the rainy and the dry season. Now we asked the question, do we see this in children in California? Now in California, we also have a wet season and a dry season, but it doesn't change the availability of fresh vegetables because they import fresh vegetables from other countries in South America during our wet season. But interestingly, when we look at children who get leukemia compared to controls, we see the same profile, indicating that children who grow up to get childhood leukemia, or the cases, look like they were conceived at a time where the mother was not eating well, or not eating perhaps enough or sufficient nutrition. So even though we don't have the wet and the dry season, it looks like people make the choice of their food quality kind of mirrors the wet and the dry season profile from this study from Africa. And this is a study, this hasn't been published yet, but we're trying to publish this, and we have some replication in other studies around the world. So this suggests that methyl donor foods around the time of conception can lead to a change in DNA methylation at this gene VTRNA2-1, affecting risk of childhood leukemia. Well, we don't know, however, if VTRNA2-1 is directly involved or just a biomarker of diet. So one final study here that shows something about nutrition or potentially in childhood leukemia is a recent study we published on twins with discordant cancers. So in this case, we didn't look at children who both got leukemia. We looked at children where one got leukemia and the other one did not. And in this case, these children are genetically identical because we only looked at monozygotic twins. So monozygotic twins, we know have the precisely same genetics. So why would one get leukemia and one not get leukemia? It may be because of unshared factors, such as different nutritional status between the two twins during development. We know that DNA methylation is critical in childhood leukemia because DNA methylation is profoundly changed from leukemias over on the left here compared to normal pre-B cells over here on the right. These twins, just briefly here, we were able to diagnose monozygotic twin pairs here from genetics, so that we only looked at the pairs who shared genetics over here on the right. We didn't find any differences between the two twins on some characteristics such as their birth weight or here the plurality order. But we did find that there were profound differences in genetics between, epigenetics between in DNA methylation between the child who got leukemia compared to their matched control. And these are some of the genes which are different in across 43 twin pairs comparing cases and controls. But the most interesting difference we saw was actually a broad demethylation in the twin who got leukemia at the time of birth. This is looking at blood spots again, compared to its matched control. So if we look at the negative coefficients, comparing twins with leukemia and their controls, we see that there's a lot more of the negative coefficients than positive coefficients. And if we look at where these coefficients occurred, they're more often in the open C region, or the broad methylated part of the genome in between genes, rather than DNA methylation at genes themselves. So this is where demethylation and perhaps genetic instability is important in these two twins, in the twin who got leukemia compared to the twin who did not. So what is the cause
Only
of this?
loyal person, because honestly,
So what is the cause of the DNA methylation differences that we're seeing? We don't know yet, but we think it's probably nutrition. Okay, and it's probably critical during this early part of pregnancy when DNA methylation profiles are established. So this is an active something we're working on actively now. Is this the window of susceptibility to environmental factors and nutrition that's most important in the cause of childhood cancer? Just the first few days of pregnancy. This is even before a woman knows she is pregnant, but it may be the most critical time for susceptibility to environmental factors that influence DNA methylation. So, yeah, just to summarize, diet is critical for healthy epigenetic development. Okay, finally, a study about genetics. So, case control studies are what I already talked about today in the presence of tobacco, is when we compare people with disease to people without disease, and we do this in studies of unrelated individuals. But I'd like to mention that we can also do studies for genetics looking at families. So this is when we see more than one presence of cancer in a family compared to over here. This is an example of a classic pedigree where we see several subjects that have cancer in a family. We can do this for childhood cancer too. And what are we going to find when we do family studies? We're going to find different genetic risk factors. When we do GWAS for genotyping for GWAS, we find these common low penetrance risk alleles. When we do family studies and we find mutations that cause cancer in family members, they tend to be these high penetrance susceptibility genes. And we know that these are causative in at least 10 to 15% of childhood cancer. And we know this from studies that have just sequenced these mutations across all childhood cancers, very large studies from, for instance, here, the St. Jude's. So when we look at family relationships in cancer, we find that there are race, ethnicity differences in risk of cancer from these familial predisposition genes. One indication of that here is when we compared in California the risk of cancer in siblings of childhood cancer cases. We see some differences by race, ethnicity. So overall, we see that the highest risk of familial associated cancer is among non-Latino Blacks in California, significantly higher than the non-Latino white population in California. And this risk is strongest for in the solid cancers, especially brain cancers, where a risk of a cancer in a sibling is sevenfold higher for solid tumors than children who are born in families that don't have a childhood cancer case. So when we look for what is the cause of that familial clustering, we find some very interesting genes, and I think this is the last slide where I have some data showing that in these families, these are Latino whites, non-Latino Blacks. We've found some very unique predisposition genes. Well, these are common ones, ATM, TP53, but other ones such as GATA2, NOD2, FLG, CBLB, these were found through exome sequencing of familial related cancer cases in California children. So I'll end there and saying a special thanks to all of the collaborators that have been involved in these studies, and thank you for your attention.
Yeah. Thank you, Dr. Joe, for the insightful presentation. And so now we open the floor for our audience to ask questions. You can raise your hand and we can start the questions now. So I've already got two questions while you were giving the presentation. So one was from Dr. Abu, he was asking about if the exposure to tobacco has been measured through a length of time. So how long had those patients in your studies have been exposed to tobacco?
Yeah, that's a good question. We assess tobacco exposure two ways. One is through a questionnaire by asking the family, yes or no, did you smoke during pregnancy, and did you smoke before pregnancy, during pregnancy, after pregnancy? What we find is when people smoke cigarettes, they tend to have started early in their life, and then they're addicted. So they actually smoked at all time periods. So the other way that we assessed tobacco was using a DNA methylation biomarker at a gene called AHRR. It's a gene that becomes demethylated in the presence of tobacco smoke, and that looking at that DNA methylation biomarker, we find a higher prevalence of tobacco than we do from the questionnaire. And we think that part of that exposure is second or third hand smoke. So it may not be that the family is actively smoking, but their household may have had exposure to tobacco in the past from smokers that is still coming out of the walls and the floors of the house and exposing the children. And so in that case, tobacco may be more important than just active smoking. It may be this passive second and third hand smoking. I hope that answers that question.
Yeah, thank you so much. Yeah, there was another question from Dr. Islam, and he was asking if you think that the infection hypothesis applies to all types of cancer or only to acute lymphoblastic leukemia.
Yes, that's a wonderful question. We have a lot of credence that infection should be involved for a cancer, which is the immune system itself, which is leukemia. It is the immune system. So it's built to respond to infection. So it's not surprising that infection causes a risk in lymphoblastic leukemia. However, we know that many cancers are related to inflammation processes in adults. So it's likely that non-leukemia cancers also have associations with infections that lead to chronic inflammation, simply because that inflammation is damaging and causes mutations in other tissues as well. But this is not studied as extensively as leukemia.
Yeah, that makes a very good sense. Yeah, so the next question from Dr. Shireen, and she's asking, how do you link congenital CMV with hyperdiploid leukemia, ALL?
Okay, that's a good question. Now, for us, what I showed for hyperdiploid leukemia was that those children have a higher presence of CMV in their tumor without having a extensive history of infection in their early life. So it's really only a hypothesis of ours that CMV may contribute directly to the leukemia. It may be that the virus itself is feeding the leukemia, and it's not so much that it changed the response to other infections. So that's only a hypothesis now. We do not have proof of that, but I believe the proof will come when we are able to treat the virus and cure the leukemia. So that will be the proof, and we are trying to do that in an animal model now. So we have a mouse model that will get leukemia from CMV, and we will then try to treat it with an antiviral drug and see if we can cure leukemia, which I think would be the proof.
Yeah, thank you so much. So I would like to remind French-speaking participants that Aurélie and Mazo will be translating. So if they want to ask in French, they can just type their question in French. So the next question goes to how you see the integration of polygenic risk score and environmental risk factor in the future to impact the clinical practice.
Right. So that's a very good question. And I realize I did not show a lot of polygenic risk score in this talk. And one reason for that is we don't have very extensive polygenic risk scores for childhood cancers yet. So the polygenic risk score in cancer story is much more complete for adult cancers, such as colon cancer, prostate cancer. In those studies with more than 100, in some cases more than 200 genetic risk factors, these can be examined as a group to identify individuals of particularly high risk of cancer. And if those families know that they have those strong risks, they can monitor their lifestyle to try to reduce the risk of cancer, specific cancers. And those polygenic risk scores, having so many different types of genes, they can look at different pathways and look at gene environment interactions with those different pathways. And I believe that will be a big theme of the PGS talks that you'll be seeing later in the series.
Yeah, sure. Agree. Yeah, so one more question from Felix. He's asking if there are, apart from folic acid, what other specific vitamins you believe are driving an inverse association, or is it like a cumulative effect with vitamin deficiency or different nutritional deficiencies?
Right. Yeah. Yes. That's a good question. I do think it's more than simply folic acid, and I haven't shown we see stronger risk effects for something called a healthy diet index. So this is a risk factor which is a combination of healthy foods rather than simply looking at vitamin supplements. Looking at a whole combination of healthy foods, there's even a stronger reduced risk of leukemia in those families. So I don't think that it's the risk effect is confined to folate. It's even the folate pathway has other critical vitamins such as vitamin B6 and vitamin B12 that all need to be in a balance to promote health. So I don't have specific information on other causative or healthy nutrients besides folate, but I do believe that we will see more being important in the future.
Yeah, so you believe that we have to look to the nutrition in a more holistic way to understand what causes this, what leads to leukemia.
Yes, I believe so. And the balance is important. We can't just flood one nutrient into our body without balancing the other ones.
Yeah. Okay, so the next and likely last question would be, how would the research finding of both environmental and genetic risk factors influence the public health policies and recommendations?
Yeah, so I think in the future, we will have we will know our risks for cancer much more deeply than we do now. And we will be able to individualize the risk prediction and prevention for individuals in our society. One thing we can also do at the public health level is screen for the early events of cancer. So the neonatal blood spots, which I showed as many times in this talk, those are used as a screening tool for inborn errors of metabolism, such as phenylketonuria or thalassemia. In California, they screen for 90 different diseases at birth. But on a public health level, maybe we can screen for cancer, early signs of cancer, the pre-leukemic mutations, and for those families, institute a targeted prevention so that their immune system will develop normally. It may be that there are some targeted interventions that are being used now to reduce risk of asthma and allergies in children who are highly susceptible. But we may be able to target those families in the future to prevent leukemia in their futures. And in many cases, it's the same targets to have the development of a healthy immune system will also promote health and reduce risk of cancer.
Yeah, thank you very much. So to conclude our presentation, what are some key skills and knowledge areas you would like young researchers to focus on? And if you want to give an advice to young researcher, what would that be?
Yeah, so for young researchers, I do think epidemiology is a growing field, with importance. So please look out for mentors who can teach and understand about population health, and those kind of opportunities to do epidemiology are at major, large research institutions, because we need populations to do that kind of science. And I would say for young researchers, the whole field is becoming more mechanistic and biology. And so it's not simply genetics. Genetics, there's a lot of there's definitely a lot of studies that are very interesting in genetics, but I think I made the point today that it's really environment that needs to be examined as well, and the two things need to be examined together. So I think for young researchers, having a strong background in biology and genetics is important, not simply try to get an array of, or a range of training opportunities, so that you can kind of make connections between risk factors and not simply look at one thing.
Yeah. Okay, so we conclude our webinar now. Thank you, Dr. Wiemels, for answering all the questions and for the very interesting presentation. So I would like, before we leave, to ask everyone to answer the post-webinar survey after we finish, so that we can, because your feedback is invaluable. So thank you again for everyone who attended, and thank you, Dr. Joe, for this nice presentation. Hoping to see you soon in more webinars.
Okay, thank you, Dr. Sarah.
Yeah, thanks to you.
Great talk. Thank you, Joe.
Oh, hi. Thank you, Felix. Good to hear from you.
Thank you all. So have a good day. Bye.
You too.
Bye.